different economic situations, the different public health support in those areas, sanitation, food, etc. In those very poor countries, there is not any significant level of sanitation, of sewage control. Clean water is a challenge. Nutritious food is a challenge. In that population, sporadic cases of hantavirus are likely to have occurred, never been diagnosed. In the United States, because there was a cluster of these cases that occurred in a relatively short time frame, the public health authorities, Utah should be in this.
That's what I'm saying. Where's Utah at?
Yeah. Since there are
That makes four corners.
four corners, you need another corner to add to it. That the public health authorities in this area recognized that the cause of disease was something that could not be identified. Centers for Disease Control was their resources were called in, and ultimately it was identified as a new hantavirus. And that, because it was a new hantavirus, it was given the name the sin nombre virus, as in honor of the Native American and Hispanic populations that were infected. Sin nombre meaning without a name, referring to the new characteristic of the virus that was identified. Transmitted by deer mice, urine, feces, and soil, aerosolization of that contaminated soil, inhalation of that dust containing the virus, and causing the pulmonary disease associated with that. Relatively rapid death. I think the death rate now, because of what we know about the symptoms of the disease, that this mortality rate has likely dropped below 35%. The video talked about an individual who had survived as a result of cardiopulmonary bypass, lungs filled up with fluid, so that the way that the blood was oxygenated is they put the patient on bypass, artificially oxygenated the blood until the fluid in the lungs cleared, and they could then resume their own respiratory function.
So that's a function of like the cytokine storm, right?
Yes.
The overreaction. Have they ever considered like giving him immunosuppressive drugs, like chemorphine, just to knock him down for a few days when they get something like that, so they can't overreact?
Well, that's already happened by the time that those patients start to develop symptoms.
So by the time they're already too far.
Yeah, there's already been too much damage due to our own responses to the virus that probably is too little too late at that point, but a good line of thought. Some bacteria associated with animals, an organism called Francisella. Species tularensis reflects the geographic area where this infections with this bacterium were first identified. This is a bacterium associated with rabbits and exposure to rabbits. Abrasions, cuts in the skin, and exposure then to either the direct contact with the rabbit or with blood from the rabbit, making entrance through those breaks in our protective barrier, our protective epithelial barrier, allows then for entrance of the bacterium. It is a bacterium, so it can be treated with antibiotics. The reason I put that there is because this is one of the organisms that is a potential bioterrorism agent. There is a group of bacteria, fungi, viruses, because of their ability to be transmitted to large numbers of individuals and then person to person, are what are called select agents, potential bioterrorism agents. and Francisella tularensis is one of those. Wild animals throughout the northern hemisphere in the United States, rabbits primarily. So protective barrier when handling rabbit carcasses to prevent acquisition of the bacterium. Other animals do harbor the organism, but not to the extent that rabbits do. So it's primarily veterinarian recreational exposure for this organism. Protective clothing, insecticides, because bites from ticks from those animals that can transmit the blood from the animal to the human, and then treatment with antibiotics is the primary mechanism. Another bacterium in the genus Brucella causes a disease called brucellosis. This is of significance in herd animals, primarily in cattle, that in the 1900s that there were large herds that became infected with this bacterium that led to transmission to humans. As a result, the agricultural practices, veterinary practices, are to screen herd populations, cattle herds, to certify them as being brucellosis free. Once that herd has received that certification, then the meat from that herd can be sold. So it's a lot of economic incentive to control infection within herd populations and to eliminate it so that meat then can be sold. Other species affect other organisms. Not of significant prevalence in the Pacific Northwest. We rarely will see any cases, particularly here on the west side. Occasionally on the east side there'll be a few cases. The only real experience I've had with this organism, or with Brucella in general, is with this organism from goats. When I was doing my clinical training in Galveston, there was a cluster of cases of Brucella melitensis infection that was ultimately traced back to goat cheese that was produced through non-pasteurized goat milk and then sold in neighborhood stores that led to infections. The situation that we were involved with at Galveston was the most challenging case, in that the antibiotic that's used to treat the infection is tetracycline. One of the infected individuals was a pregnant woman in whom you cannot use tetracyclines for treatment. So there was a large amount of challenge in terms of infectious disease physicians and developing appropriate therapy.
Which I want to do.
Pardon me?
What they want to do.
There was a 2 antibiotic combination, and I couldn't tell you what it was at this point. That was more than 10 minutes ago. Prevention, pasteurization of milk, and inspection and certification of deliveries. In terms of transmission by other animals other than the ones we've just previously described, other mammals, arthropod transmission of both bacteria, viruses, and protozoa can occur. There's a group of bacteria that has the same characteristic as the chlamydia, and that they are obligate intracellular pathogens. In fact, this group of organisms called the rickettsias are the simplest genetically of any of the bacteria. They've got the smallest genome because they have specialized to a intracellular symbiotic type of existence with eukaryotic cells, where some of their cell function is supported by that infection. These are the organisms based on our 3 biological domains. In the first domain, the bacterial domain, partway up there was the cyanobacteria and the chloroplasts. Off on the other side, about halfway up that domain, are the rickettsias and another organelle, mitochondria, with the assumption that mitochondria developed from an intracellular association of rickettsias with another organism that ultimately evolved into eukaryotic cells, with the rickettsia becoming specialized for the function of mitochondria. What do mitochondria do in eukaryotic cells? Generate ATP. Okay, so they're basically an ATP producing factory. Transmitted by blood-sucking arthropods, either lice or ticks. 3 different groups: the typhus group transmitted by lice, the spotted fever group by ticks, and another group that we call the Ehrlichia is also transmitted by ticks. Don't worry about the detail of these different groups. Understand that the ricketsias are obligate intracellular pathogens, that they are arthropod transmitted. They are bacteria, so they can be treated with antibiotics. But again, the challenge is diagnosing the disease, because these diseases tend to be relatively nonspecific. They have a constellation of symptoms that are referred to as a flu-like syndrome, with the exception of spotted fever. There are some symptoms of that that are relatively distinct. Early diagnosis and treatment with antimicrobials prevents long-term effects of these organisms. This is the lice that transmits our typhus group. This shows the geographic range of our second group, the spotted fever group. You notice about the geographic distribution of the ricketsias that cause Rocky Mountain spotted fever.
There's very little in the Rocky Mountains.
Not so much in the Rocky Mountains.
It's on the East Coast.
That's where the disease was first identified. And then once the methodologies for identifying the organism, the symptoms of disease were associated with the clinical presentation and the modes of transmission, it was found that the central and eastern seaboard had a much higher prevalence of spotted fever. There is actually a CDC rickettsial disease laboratory in Hamilton, Montana that was established to study the rickettsias and the spotted fever rickettsias. And it was a result of that study that led to this sort of recognition of its geographic distribution. Spotted fever presentation. Because the organism is transmitted in the blood, it causes a effect on our clotting cascade that results in microhemorrhages, microclots. Those clots then lodge in capillaries that then cause localized necrosis Each of those red spots is an area where the tissue is starting to die because of lack of support from the bloodstream due to the microclots in the capillaries. Fever and then this rash presentation typically will lead to a diagnosis of spotted fever. Because these are intracellular organisms, we don't culture them in the lab. You could use a special type of cell culture technique, but it takes a relatively long period of time. So diagnosis for all these organisms is immunologic. That is, we look for antibody in our blood against the particular organism. So once there's a diagnosis of potentially spotted fever, then blood is collected and sent to an immunology lab to identify antibody against that organism. It's referred to as serodiagnosis, serologic diagnosis based on presence of antibody. And then at that point, diagnosed disease can be treated with antibiotics. Because again, the rickettsias are bacteria, so they're susceptible to antimicrobials. Another arthropod transmitted bacterium transmitted by ticks producing the Lyme disease